Chapter 12, Section 2, Ribosomes and Protein Synthesis. Learning objectives: explain how the genetic code works, describe the role of ribosomes in assembling proteins, and understand how molecular biology relates to genetics. The first step in the process of decoding genetic messages is transcription, which is the copying of a nucleotide-based sequence of DNA into mRNA. The next steps lead to the assembly of a protein. Proteins are made by joining amino acids together into chains called polypeptides. 20 different amino acids are commonly found in polypeptides. The specific order in which amino acids are joined together in a polypeptide chain determines the shape, the chemical properties, and ultimately the function of the protein. How is the order of bases in DNA and RNA molecules translated into the order of amino acids in a polypeptide? 4 bases of RNA form a kind of language with just 4 letters, A, C, G, and U. We call this language the genetic code. How can a code with just 4 letters carry the instructions for 20 different amino acids? The genetic code is read 3 bases at a time. Each word of the code is 3 bases long and corresponds to a single amino acid. This 3-base word is known as a codon. A codon consists of 3 consecutive bases that specify a single amino acid to be added to the polypeptide chain. Because there are 4 different bases in RNA, there are 64 possible 3-base codons in the genetic code. Most amino acids can be specified by more than one codon. For example, 6 different codons UUA, UUG, CUU, CUC, CUA, and CUG specify for leucine, but only one codon, UGG, specifies for the amino acid tryptophan. Start at the middle of the circle with the first letter of the codon. Move out to the second ring to find the second letter of the codon. Then find the third and final letter among the smallest set of letters in the third ring from the center. Next to the third letter is the amino acid that the codon specifies. Whether a message is communicated in writing or the genetic code, it needs punctuation marks. In English, punctuation tells us where to pause, when to sound excited, and where to start and stop a sentence. The genetic code has punctuation marks too. The methionine codon AUG also serves as the initiation or start codon for protein synthesis. Following the start codon, mRNA is read 3 bases at a time until it reaches one of 3 different stop codons, which end translation. At that point, the polypeptide is complete. You can think of the sequence of bases in the mRNA molecule as a set of instructions. The sequence gives the order in which amino acids should be joined to produce a polypeptide. Once the polypeptide is complete, it then folds into its final shape or joins with other polypeptides to become a functional protein. If you ever assembled a complex toy, you know that just reading the instructions is only the first step. You need to follow the instructions to put the parts together. A cell part that acts like a tiny factory, the ribosome, carries out the assembly tasks. Ribosomes use the sequence of codons in mRNA to assemble amino acids into polypeptide chains. The decoding of the mRNA message into a protein is a process known as translation. Translation begins when a ribosome attaches to the mRNA molecule in the cytoplasm. As each codon passes through the ribosome, several molecules of tRNA bring the proper amino acids to the ribosome. One at a time, the ribosome then attaches these amino acids to the growing chain. Each tRNA molecule carries just one kind of amino acid. In addition, each tRNA molecule has 3 unpaired bases that are together called an anticodon. Each anticodon is complementary to a codon of mRNA. In the case of tRNA molecule for methionine, the anticodon is UAC, which pairs with the methionine codon AUG. The ribosome has a second binding site for tRNA molecules for the next codon. If the next codon is UUC, a tRNA molecule with an anticodon of AAG fits together against the mRNA molecule held in the ribosome. That second tRNA molecule brings the amino acid phenylalanine into the ribosome. Like an assembly line worker who attaches one part to another, the ribosome helps form a covalent bond, called a peptide bond between the first and second amino acids. In this example, there are methionine and phenylalanine at the same time. The bond holding the first tRNA molecule to its amino acid is broken. That tRNA then moves into a third site from which it exits the ribosome. The ribosome then moves to the next codon, where tRNA brings it another amino acid. The polypeptide chain continues to grow until the ribosome reaches the stop codon on the mRNA molecule. Then it releases both a newly synthesized polypeptide and an mRNA molecule. The 3 major forms of RNA, mRNA, tRNA, and rRNA, are each involved in the process of translation. The mRNA molecule carries the coded message that directs the process. tRNA molecules deliver the amino acids, enabling the ribosome to read the mRNA's message and to get translation just right. Ribosomes themselves are composed of roughly 80 proteins and 3 or 4 different RNA molecules. These rRNA molecules hold ribosomal proteins in place and carry out the chemical reactions that join amino acids together. RNA molecules not only carry the genetic code, they also play a key role in translating it. Gregor Mendel might have been surprised to learn that most genes contain nothing more than instructions for assembling proteins. He might have asked what proteins could possibly have to do with the color of a flower, the shape of a leaf, or the gender of a newborn baby. The answer is that proteins have everything to do with these traits. Remember that many proteins are enzymes which catalyze and regulate chemical reactions. A gene that codes for an enzyme to produce pigment can control the color of the flower. Another gene produces proteins that regulate patterns of tissue growth in a leaf. Yet another may trigger the female or male pattern of development in an embryo. In short, proteins are microscopic tools, each specifically designed to build or operate a component of a living cell. After scientists learned that genes were made of DNA, a series of other discoveries soon followed. When they explained the genetic code, a new scientific field called molecular biology had been established. Molecular biologists seek to understand living organisms by studying them at the molecular level, using molecules like DNA and RNA. Molecular biology provides a way to understand the links between genes and the characteristics they influence. One of the most interesting discoveries of molecular biology in the near universal nature of the genetic code. Although some organisms show slight variations in the amino acids assigned to particular codons, The code is always read 3 bases at a time, always in the same direction, and is always translated on ribosomes composed of RNA and protein. Despite their enormous diversity in form and function, living organisms display remarkable unity at life's most basic level, the molecular biology of a gene.